it gives me great honor to moderate the infectious disease section now for the conference and introduce the next speaker for the day, who is Naveen Vankadari from the University of Melbourne in Australia, who will be presenting on the genetics of the human TMPRSS2 regulates COVID-19 infection, and Arbidol as a potential drug in COVID-19 patient viral clearance. As we say in Kenya, Karibu Naveen.
Thank you, Pranaraj. Thanks for introducing. Let me share my screen. Can you see my slides?
Yes, we can.
Okay, awesome. Foremost, thank you for introducing, and as well as I thank Beyond Science Initiative for inviting and getting an opportunity to speak in this forum. I would like to simplify my talk in a very generous way and how human mutations or human genome play an important role in the COVID-19 infection. In one such gene is involved in human genome is called as TMPRSS2. So before going more into the topic, I would like to tell tell in a simple and animated from the WHO that in the race of the COVID-19 variants, there are some variants which is running faster, or they are ahead of rest of the variants, which is involved in generating so many serious variants, which we all facing, like an Omicron, Delta, Mu, and whatever it could be. So what is happening is mutations. So mutations are the changes in the genome which is taking place, and some of the COVID-19 variants is resulting into the faster growth and faster infection compared to the rest of the viruses. So along the same line, if you look into a trajectory of the virus from the animal source to a worldwide pandemic, and then still, despite of so many vaccine strategies and vaccines are available, both mRNA-based vaccines, live attenuated vaccines, still we are looking for a potential therapeutics to treat the infected patients. Using a electron microscopy is a powerful technique or a tool which can image the inceptiotemporal, which is called as where we can capture the time and space of the virus infecting in a human cell. So let's look into this, a small picture of the interface of the human cell where the virus of the SARS-CoV-2 interacting. It can interact with so many number of a different host factors sitting on the surface for subsequent entry of the virus. By now, we all know that ACE2 is one of the primary receptor for cellular entry. However, when you look into an transmission electron microscopy and as well as scanning electron microscopy versus the virus in the host cell, a single host cell can be attacked with hundreds and sometimes probably thousands of viral load. So it all related into a viral load, and then how the genome is transitioning, how the individual patients or individual human beings are being responding for a different viral and vaccines as well. So in that regard, in the race of a COVID-19 or race of an evolution of an SARS-CoV-2 viruses from a single originated virus from Wuhan to so many different variants what we have, there is a progeny or prolongation of a different variants like an epsilon to Omicron what we have, and some more variants are coming, and XBB and other things as well. So what's happening is all these variants, despite whatever the lineage is, they're all infecting the humans, we know that, and how the individual humans are responding towards it, and how the genetics of the human are going to manipulate this virus as well for the infection rate. So before going deep, so most of the therapeutic options at the moment is involved in the spike protein, which is one of the key protein involved in the virus engaging in our interaction with the host cell. So when we look into the cryo-electron tomography more, so the right side panel of the video will show the three-dimensional cryo-electron tomography. The spike protein is not just rigid what we are seeing in the textbooks or in news like in one conformation, but it exists in so many different conformations as well. So making it monoclonal antibodies or nanobodies or any polyclonal antibodies specific to some region is making very challenging and as well as not efficient economically as well, because spike protein is changing constantly, so many variants, all the antibodies not effective, treatment for the antibody itself is so expensive. So that is one of the reasons for a potential flaws or for the antibody treatment in many of the viral infections as well. So if you look into the lineage or what could be the cause for a viral infection, is so many factors are there. One is the viral load, the host factors, and as well as environmental factors. If you look into the host factors, which I'm going to talk mostly about, we think about the gender, age, and so many, and the pre-existing conditions of the body, and so many other factors are also there. But I'm talking today about one of the factors called as ethnicity. So this research is based on the observation. A good science and a good research is always starts from a good observation. In early 2020, we noticed that compared to different ethnic background, from European or Indian, Asian, and other ethnic background, in Indian population, despite the viral load is high, the severity of the infection was mild in some patients. Then question is why? And what is making in the human genome is causing such kind of variations in the infection compared to different ethnic background is one of the other host factor. So in that regard, we collaborate with the AIG hospital in India, in Hyderabad, and we recruited nearly 1,000 patients who has been, or 1,000 people who just admitted for thinking about the COVID-19. Among them, about 50% are in 510 patients were COVID-19 positive and confirmed with the PCR techniques and clinical symptoms and all the blood reports. And then what we did is we did a whole genome sequencing and basically exome sequencing of all the healthy and the infected patients, and look for what are the gene changes happening between the healthy and the infected patients, and what genes are involved in human is reflecting back into the difference in the sensitivity or viral infection, or why some patients are more infected or are more severe condition compared to the other things. Fortunately, we could able to find a gene called TMPRSS2 in human. A single point mutation in the human genome has resulted in that kind of a mutation, which changed the result in the different level of infection. So let's see what is happening here. So there are three types of symptoms: asymptomatic, mild symptomatic, and severe symptomatic. So a patient who has a very small lung lesions are observed, which is an asymptomatic. The patient is infected, but not much lung lesions are there. But some patients are being infected with moderate lung lesions are there in CT scans, and some patients are come out with a severe infection. Fortunately, a patient who has a mutation in the gene called TMPRSS2 in human, called valine 162 methionine, has given a protection, which means like a patients who has a mutation are asymptomatic to moderate, but the patients who do not have this kind of a mutation are more severely infected, which means like mutations in the human genome protects against the COVID-19 infection. We may think that a single point mutation, how it can be so profound role. If you look into that, our hair color is single point SNP, a single nucleotide polymorphism. Eye color is SNP. Lactose intolerance is SNP. Skin color is SNP. So many gene and genetic variants in the body, which has been very well established, showing that a single SNP can completely change the whole cascade of viral infection or any genetic phenotype changes as well. This is one of the example as well. So we looked into the gene production of the TMPRSS2 wild type and the mutant protein and see how it is going to play an important role in the infection as well. And we know that the V160M mutation is going to change or reduce the infection in human patients, but how? That's a question. So we would like to answer always.
Naveen, sorry to interrupt. Could you try to rush through the presentation due to time constraints?
Yeah, sure. So we solve the structure of a TMPRSS2 protein and show that when the mutation is occurring, that human protein getting distorted, that's giving the stability of the TMPRSS protein. And that's also we found in a mouse longevity. When the mutation is happening, the mouse is going to live longer. And as well as we developed a couple of a key important drugs which can block the TMPRSS2 function and which can increase the longevity in the human patients as well, and thus acting as a potential therapeutics from the single gene mutation as well. Thank you, and I'm happy to take questions.
Thank you, Naveen, for that really insightful presentation, which gives a new dynamic approach to COVID-19, treatment approaches and more options. There's a question in the Q&A section, which is just what causes mutation to the V160M? Maybe you can answer that and then stay online to answer the rest on the chat section.
Okay, sure. So the cause for the V160M mutation is a genetic diversity. It's not any specific influence, like how lactose intolerant. Some people are lactose tolerant, some people are lactose intolerant. So over the course of time, some mutations are going to happen. So which is required and non-required things. So that's just genetic makeup in the human genome.
Thank you, Naveen, once again.
Can I just jump in with a quick question?
Yeah.
So that's a great presentation, Naveen. And I'm just wondering, how important do you think, as the virus mutates, is this particular molecule going to be? Do you think there's going to be, we've seen with various mutations how the viral entry is changed. Do you think this is going to be something that the virus might actually circumvent?
No, because we are not targeting the virus. We are targeting the host gene. And this region is not at all involved in all the variants is happening in the receptor binding domain, the S1 domain of the spike. And this is not going to interact with the S1 domain. It is going to interact with the cleavage site of the spike. So it is always there as long as SARS-CoV-2, it's going to work well.
Thank you.
Thank you. Don't know if there's any other question, but Naveen, thank you. We appreciate you joining from Australia, and we hope you can stay on for the rest of the conference.
Thank you, Pranav.
Okay.